In this video, we'll study the functional correspondence between one gene and one protein. What lessons have we learned from Mendel's and Morgan's experiments? A gene is an element of hereditary information that is responsible for the manifestation of a specific trait and the modification of a specific trait upon mutation. However, there was a restriction to this argument. The relationship between a gene and a specific phenotype had still not been fully explained at the time. How can we study this relationship? By using a bottom-up approach, we can identify a change in a trait as a starting point and then go back to the gene to see what has caused this change. Scientists' understanding of biochemical pathways suggested that some traits could result from synthesizing a specific set of proteins, such as enzymes responsible for pigment synthesis that define eye color. A functional correspondence between a gene and a protein can also be established by studying hereditary diseases. Alternatively, we can study the correction of a mutant phenotype that was created for an experiment. Let's start with an analysis of certain hereditary diseases. The first disease, which was studied by Garrod in 1902, is called alkaptonuria. It's an inherited metabolic disease that's characterized by an accumulation of homogentisic acid in urine. Patients who suffer from this disease experience a darkening of their urine when it's exposed to air. This disorder has a high incidence in the offspring of two cousins. When Garrod studied the transmission of the disease, he noticed that it followed the laws of Mendel and that the transmission mode suggested a recessive mutation carried by a single gene. How does this accumulation of homogentisic acid happen in patients with alkaptonuria? Well, at the time, the metabolism of tyrosine was known. Tyrosine is produced by a modification of another amino acid called phenylalanine. Tyrosine is converted, either by being used or degraded, into p-hydroxyphenylpyruvate. This is then converted into homogentisic acid. Under normal conditions, homogentisic acid is then degraded into a series of intermediates. This is done until it is fully degraded into fumarate, which enters the citric acid cycle. The accumulation of homogentisic acid is linked to the functional loss of an enzyme called homogentisate oxidase, which converts homogentisic acid into formaldehyde acetoacetate. This means that alkaptonuria patients have a gene mutation that, due to its inherited nature, leads to the production of an inactive enzyme. An accumulation of homogentisic acid indicates the abnormal metabolism of tyrosine. It is not a gradual process that occurs over a lifetime. Rather, the metabolic process is altered from birth. So, in alkaptonuria patients, a functional correspondence exists between a gene and an enzyme. The second analysis is a study conducted by Fölling in 1934. Fölling studied a disease that you probably know more about, phenylketonuria, or PKU. PKU is an inherited metabolic disorder characterized by an accumulation of phenylketones in the urine. The most distinct symptom in patients with PKU is that their urine turns olive green in the presence of iron chloride. If left untreated, PKU leads to intellectual disabilities in the patient. When Fölling studied the transmission of this disease, he found that it followed the laws of Mendel and suggested that it resulted from a recessive mutation carried in a single gene. Since PKU is a metabolic disorder, we need to revisit one of the metabolic pathways, in this case, phenylalanine metabolism. In fact, we covered it in the previous example, where we described how phenylalanine is converted into tyrosine. The accumulation of phenylketones in PKU patients is due to an abnormal phenylalanine metabolism. In healthy individuals, phenylalanine is either consumed or converted into tyrosine. Following this, tyrosine is either consumed, incorporated into proteins, or degraded. In PKU patients, the conversion of phenylalanine into tyrosine is blocked because the enzyme that catalyzes the reaction, phenylalanine hydroxylase, is inactive. As a result, phenylalanine accumulates and is then converted into phenylketones. Once again, this study highlights a functional correspondence between a gene and an enzyme, the gene that is associated with PKU and the phenylalanine hydroxylase enzyme. By studying auxotrophic mutants, we can also study the correspondence between a gene and a protein. An auxotrophic mutant is an organism that cannot synthesize a specific biomolecule. In 1940, Beadle and Tatum studied the fungus Neurospora crassa. They showed that a wild-type strain of Neurospora can grow on a synthetic medium that contains a carbon source, a nitrogen source, and some vitamins. This means that these wild-type strains can synthesize all their required amino acids. However, if they took this wild-type strain and exposed it to X-rays, they could induce random mutations and select auxotrophic mutants. Some of these mutants could not produce their own arginine, meaning they needed to be grown on a minimal medium supplemented with arginine to survive. At the time of Beadle and Tatum's experiments, the arginine biosynthetic pathway was well established. Scientists knew that each step in this pathway was catalyzed by an enzyme. It starts with arginine's precursor, N-acetyl-ornithine, which is converted into ornithine. This reaction is catalyzed by the enzyme acetyl-ornithinase. Ornithine is then converted into citrulline, which is catalyzed by ornithine transcarbamylase. Citrulline is converted into argininosuccinate, and this step is catalyzed by argininosuccinate synthase. Finally, argininosuccinate is converted into arginine, which is catalyzed by argininosuccinate lyase. After Beadle and Tatum induced mutations in their experiment, they isolated 4 types of arginine-, or arg-, mutants. Each mutant strain carried a mutation in a distinct gene. Beadle and Tatum demonstrated this by using pairwise complementation assays. Before discussing the assay setup, it's important to know the life cycle of Neurospora crassa. It involves 2 stages: the haploid stage, where the organism has a single set of chromosomes, and the diploid stage, where 2 haploids, each with 2 complete sets of chromosomes from each parent, come together to form a diploid organism. Knowing this, let's return to the complementation assay. Let's consider a wild-type strain with an arginine plus or arg plus phenotype. Let's assume that the synthesis of arginine requires 2 functional enzymes, A and B, each coded by distinct genes A and B. Mutations that lead to the arginine minus phenotype can occur in either gene A or gene B, as both enzymes are required for the arginine plus phenotype to be present. To carry out a pairwise complementation assay, Beadle and Tatum selected 2 haploid strains that are arginine minus. One strain carried a mutation in gene A, while the other strain carried a mutation in gene B. They then created a diploid strain. This diploid strain had 2 wild-type copies of both genes A and B, each derived from one of the haploid parents. Thus, these strains would be arginine plus because they express the functional proteins A and B. By bringing together 2 haploid genomes and switching from a haploid arginine minus phenotype to a diploid arginine plus phenotype, the complementation assay indicated that the mutations must be on different genes. Contrastingly, if the mutations were carried on gene A in both haploid strains, the resulting diploid organism would contain 2 copies of the mutant gene A. This would produce a non-functional protein A and would remain arginine minus. Now that we know how Beadle and Tatum selected their 4 distinct arginine minus haploid strains, let's discuss their experiments. Beadle and Tatum then tested the ability of haploid wild-type and mutant strains to grow either on minimal media supplemented with arginine, or minimal media supplemented with intermediates in the arginine biosynthetic pathway, which are ornithine, citrulline, and argininosuccinate. None of the haploid mutant strains could grow on a minimal medium, but they could grow when the medium is supplemented with arginine, indicating a blocked step in the arginine biosynthetic pathway. The wild-type strain was able to grow on all media types. Next, Beadle and Tatum wanted to know which step in the arginine pathway was blocked in each mutant. To do this, they added arginine precursors to the growth medium to see which precursors restored growth. The addition of the precursor that restores growth will, therefore, determine which step in the pathway is blocked. Mutant 1, when provided with only ornithine, could grow. This indicated the inactivation of acetyl-ornithinase. Mutant 2 required citrulline. This suggested a block between ornithine and citrulline, so the inactive enzyme must be ornithine transcarbamylase. Mutant 3 grew with argininosuccinate or arginine, but not with citrulline. This indicated a block between citrulline and argininosuccinate, which meant that the non-functional enzyme was argininosuccinate synthase. Mutant 4 grew only with arginine, which confirmed a block between argininosuccinate and arginine. This meant that the non-functional enzyme was argininosuccinate lyase. Identifying which precursor restores growth allows us to determine which step in the pathway is blocked. Mutant 1 requires ornithine for growth, therefore the acetylornithinase gene must be inactive. The same can be seen for the other mutants. In conclusion, each mutated gene was responsible for synthesizing a specific enzyme, and a mutation in the gene caused the synthesis of an inactive enzyme. This experiment further supports the one gene, one protein hypothesis, establishing a correlation between a gene and a protein.